One is online. Are you still hearing me? I just got a buzzy sound. Okay, good. All right. Announcements, right. There is a tomorrow, there is another senior seminar on human papillomavirus, cervical cancer, and the development of vaccines. So it's sort of topical. Yazid and Linan will be presenting tomorrow, CST 300 at 4 p.m. and there's also a Zoom link, which I believe is on today at Earlham. But if you, I can send you that information if you need it. That's tomorrow. And then on Thursday, there is another senior seminar about urban ecology, animals and plants in urban settings. Pretty interesting. There was a recent study about how birds, during the COVID shutdown, bird song became more elaborate and didn't use as much volume. Used less volume but more tonalities that developed very quickly when cities became more quiet during the COVID lockdown. So that happens very fast. I don't know if I'll talk about that, but it was an interesting thing in Science magazine just this week or last week. So those are two announcements. I have your quiz. Your grades are on Moodle. I can give them to you after class. Class today will not be 3 hours. Class today might be as little as an hour and 2 minutes. We'll see. So we're going to pick up where we left off, talk about the liver, do a little review of anatomy, which you folks did pretty well on the quiz, so I can go through quickly on that. Talk about blood flow in the digestive tract, which is complicated, and we'll talk about it. So the liver, let me just skip ahead a couple of slides. I'm sorry about these being out of order. You know the liver, and you know some of the plumbing. You know quite a lot of the plumbing, bile duct, common bile duct, hepatic duct, right hepatic duct, left hepatic duct, common hepatic duct, bile duct, cystic duct, right? Some of the plumbing from the liver to the digestive tract. But now I want to talk about some things other than bile. So we're going to review the anatomy. We're going to talk about the blood flow and the bile flow. Then we're going to talk about the intestines. Anatomy of the intestines, blood flow to the intestines. Where is that? Blood flow to the liver and to the intestines, and how bile is important for the digestion of fat, and the molecular mechanisms for the absorption of sugars from carbohydrate and amino acids from protein. That's where we're going for the next hour. So the top 11 functions of the liver, which you do not have to memorize, but you probably should know some of them, and some of them you already know. You know that it secretes bile and bicarbonate for digestion of fat and neutralization of acid. And now let's talk about some of the other important mechanisms of the liver that we're not going to dwell on. But in order to be honest about the treatment of the liver, I at least have to mention these. It stores calories as glycogen. So your liver has glycogen deposits. Glycogen is a starch. So potatoes store calories as starch. We store calories as glycogen. Glycogen is not glucagon. Okay? Remember, glucagon was a hormone. It's a protein. It's a hormone. Glycogen is a storage polysaccharide, a starch. So it's important. The liver is a storage place for the storage of calories between starvation events. Sort of like fat is also a place where we store calories. It turns amino acids into fatty acids, which we need for making and regenerating our membranes. When we are starving, it turns glycogen into glucose to help provide energy to our brain and the rest of our body. So when we are fat and happy, we are storing glycogen in the liver, and when we are starving, we produce glucose from that glycogen. And we don't even have to be starving, we can just be fasting between meals. Now some other things. Angiotensinogen, which you learned about in the context of the renin-angiotensin-aldosterone story. Angiotensinogen comes from the liver. There are other hormones that come from the liver. It is the source of cholesterol that you need for your cell membranes. And people who have high blood cholesterol, who take a statin drug, statins are drugs that inhibit the production of cholesterol because their blood cholesterol is too high. The target of that are the enzymes that make cholesterol in the liver. So for example, I am taking a drug called Lipitor, or atorvastatin, because my blood cholesterol is a little too high. And the doctor said, let's see if we can improve that. So I'm taking Lipitor to inhibit an enzyme in the production of cholesterol. Urea, that we learned about in the context of the kidney, is made in the liver from metabolism of protein. Albumin, the protein in blood that you learned about in the context of the kidney, is made in the liver. Red blood cells are degraded there, as well as in the, well, there are other locations for the degradation of red blood cells, but the hemoglobin, the heme in hemoglobin, which your membrane, your red blood cells are full of, gets converted into that green pigment, green bile pigment. It's an organ for clearance of certain toxins and metabolites. So for example, some of the drugs that you might take, like, okay, let's talk about Lipitor. Lipitor is broken down, not only does it have an effect on the synthesis of cholesterol, it is broken down, I think, by the liver and excreted in bile. So its clearance depends on liver function. And it also, the factors in your blood that are important for your blood to clot when you have an injury are produced in the liver. People who have hemophilia and have a bleeding disorder, it's because they have a defect in the production of these clotting factors. It's a genetic defect in the production of clotting factors in their liver. So a lot of functions. You do not have to memorize these all. We are going to focus on the bold one at the top. But you should know some of them. Because I could ask you, give me 6 of the important functions of the liver. And if you only know the first one, it's only 1/6 of what I'm asking. Okay, anatomy, you folks are good at this. Questions about this anatomy? So I'll just move right on. You showed me a lot of knowledge on the quiz.
What happens when the gallbladder is removed? It just strips straight into it.
When the gallbladder is removed, the bile from the liver drips straight into the duodenum. It means that the storage of bile is impaired, and so people who have had a gallbladder removed are typically told to eat a low-fat diet, because if they eat a big bolus of fat and we're only dribbling in a little bit of bile, that can cause a problem with fat digestion, and it typically causes diarrhea and other digestive insults. They can digest fat, but they are told, do not eat a meal that has a lot of gravy or, a lot of butter or something. Eat a low-fat diet, or else and they probably develop a pretty intrinsic knowledge. Okay, can't eat more than that because I'm going to feel bad in a couple hours if I eat more than that much fat. So we need to talk about blood flow. And we're going to talk about it in two events here in this class. First, we're going to talk about blood flow to the liver. And then we're going to talk about blood flow to the stomach and to the other digestive tract. So let's stay focused on the liver for right now. So let's get oriented. Abdominal aorta, stomach, lesser curvature, greater curvature, lower esophageal sphincter, cardia, pylorus sphincter is behind there. Here's the gallbladder of the liver, which has been sort of moved out of the way. I don't know how they did that, so this is a drawing. Blood flow, aorta, celiac trunk. I think I gave you the slide, but you probably can't read it. Celiac, just like the word celia, only with a C at the end. Or celia, the name, celia, with a C at the end. Celiac. It's going to split into several arteries, and the one we're going to talk about now is called the common hepatic artery. It means it goes to the liver. This branch to the right, common hepatic artery, and then the proper hepatic artery after it splits, is bringing arterial blood to the liver. The liver is, metabolically active. It needs arterial blood, and that's how arterial blood gets to the liver. The liver also processes venous blood because the products of digestion, whether it's from the stomach or from the intestines, goes to the liver through venous circulation. So right now we have to know the name, the portal vein. Right now we need to know that the portal vein brings venous blood from the stomach and the intestines to the liver. So all those nutrients that this blood picks up from the stomach and from the intestines, and we're only seeing the large intestine, but you know that the small intestine is here too. All that nutrient-rich blood goes to the liver first. And so carbohydrates, sugars in that blood get turned into glycogen. Amino acids in that blood get turned either into fatty acids or into urea. Or both. Fats in that blood, well, let's leave fats aside. So the liver is receiving both arterial and venous blood. And it's essential that it receives both by these pathways. Any questions about this anatomical confluence of arterial and venous blood? The arterial story, celiac trunk, common hepatic artery, proper hepatic artery going to the liver, and venous blood, the portal vein, taking venous blood to the liver. Question about that? Okay, so now we have to talk about the cellular structure of the liver. It's pretty interesting. So let's just walk through this image. Here's your liver. Here's a piece of the liver blown up. And it has these hexagon-shaped structures called lobules. This is where the hepatocytes live. The lobules are perfused with both arterial, in red, and venous blood. So here is a blow-up of a single lobule. Here is a branch of the hepatic artery, the proper hepatic artery. This is not the proper hepatic artery, it's a branch of it. But it's bringing arterial blood to this lobule. Here is a branch of the portal vein, bringing venous blood to this lobule. Let's not worry about the bile yet. So, and the cells in the lobule, these brown cubic shaped things, are the hepatocytes. Now, you know that ordinarily, when we have exchange happening at the cellular structure, we typically have capillaries that are doing that. There's a capillary that's lined with endothelium and an interstitium between the capillary and the tissue that is receiving the blood. That is not the case in the liver. In the liver, the arterial blood and the venous blood sort of percolates in spaces between the hepatocytes called sinusoids. Sinusoids. Sinus means space or something. Oids means like. So we do not have capillaries. We have the blood coming in direct contact with the hepatocyte because it's surrounded by these sinusoids that contain mixtures of arterial and venous blood. I used a word there, percolation. So if you think about, if you get a rainstorm and the water is moving into the soil, it's moving around the rocks and it's moving around the sand and it's moving around the blobs of soil, that's percolation. When you make coffee and you pour hot water on a bed of coffee grounds, the water is percolating through the coffee. That's the process of percolation. Here, the blood is percolating around the cells of the liver, the hepatocytes. So the blood is coming in, the arterial blood and venous blood from the portal vein are coming in, percolating around the liver cells. Nutrients are being absorbed by the hepatocytes. Toxins are being maybe delivered to the venous blood. Some of the toxins are going to go into the bile, which we'll talk about in a minute. That percolation, then the blood percolates to another vein called the central vein. This is in the center of a lobule. So venous blood in through the portal, a branch of the portal vein, and venous blood out through a central vein. Here is an actual histology of it. So here is, the hexagon helped us see the lobule. Here's the central vein of the lobule. Here is a branch or a venule from the portal vein. Here is an arteriole from the proper hepatic artery. Let's not worry about the bile ducts. They're there too. So blood is coming in through the portal vein, the venule of the portal vein, from the arteriole branch of the hepatic artery, and blood is now percolating through these white spaces, which are the sinusoids surrounding these red and purple hepatocytes. Nutrient exchange for arterial blood, nutrient exchange for venous blood. Hepatocytes are doing their good work, and then the blood percolates to the central vein. The central vein is going to drain into the inferior vena cava. The vein that you've heard of. Actually, the central vein is going to drain into the hepatic vein, which is going to drain into the inferior vena cava. We'll see that in a minute. Key concept, sinusoids, not capillaries. Key concept, both arterial and venous blood coming into the functional unit and then venous blood draining out. Yes?
How does that necessarily happen if they're both like percolating together? How can the liver decipher like?
It's just taking what it needs, right?
Okay.
So from the arterial blood, it's getting oxygen.
So all of it becomes venous.
It all becomes venous in the end, but it takes the oxygen it gets from the arterial blood, and it takes the nutrients from the meal that came to it from the stomach and from the intestines from the venous blood. And then it all becomes venous blood and goes into the hepatic vein, into the inferior vena cava, to the heart, etc. One of the important functions of the liver is to secrete bile. You know this. And so again, there are these little ducts, ductules. They've got a funny Italian name, canaliculi. Let's just call them ductules, where the liver hepatocyte secretes bile into the ductules, which collect into ducts, which are going to connect to the ducts that have names. Hepatic duct, common hepatic duct, common bile duct. If the sphincter of Oddi is closed, the bile backs up into the gallbladder. If the sphincter of Oddi is opened, which happens because of the hormone CCK, so the thing that opens the sphincter of Oddi causes contraction of the gallbladder, and we squirt bile into the intestines. But the point is, the hepatocytes make bile, secrete it into bile ducts, which flows into either the gallbladder or into the duodenum, depending on whether the sphincter of Oddi is open or closed. So that's the liver from sort of an anatomical perspective. Both blood flow anatomy, I haven't told you about some of the mechanisms yet, but from a blood flow anatomy and from a cellular anatomy, that's the liver story. Now we're going to talk about the anatomy of the intestines. And then we're going to come back and talk about molecular mechanisms for digestion. So I'm putting this out anatomically first. Anatomy of the liver, gross anatomy, microscopic anatomy. Now we're going to get to the gross anatomy of the intestines. So this is a cartoon of the intestines of a normal abdomen. You didn't see a normal abdomen on this cadaver because the liver had pushed all of this so far down that we couldn't even see it until we sort of pulled it up out of the pelvis. But this is a more typical view. You would typically see the small intestine wrapped, well organized, and surrounded by the large intestine. The yellow area that we're seeing here just in little bits is the mesentery that you did see in lab. This area here that has been sort of cut off would be the omentum that is attached both to the greater curvature of the stomach and to the transverse colon that sort of drapes over the small intestine. I don't think you saw that very clearly in the lab either. So let's talk about some of the gross anatomy of the intestines. And so in the past I have played this game. Let's play this game a little bit. Let's start in the back. How about Easton? You ready to go? What's this pointing to?
So.
Good. Now in the lab, if you gave me stomach, if I pinned this right here, what other information would you give me that was higher level than just stomach? Body, serosa. If it were pinned right here, you could say that's the lesser curvature, right? So I'm just reinforcing that in the practical quiz, which you're going to take on Thursday night or Friday afternoon, you want to give me high-level information. This is just doing those simple things, so don't get deceived because we're doing simple things here. Okay? Okay, who's next? Michael. What's this one? Duodenum. Okay, Olivia. Let's skip this one. What's this one? This one.
I was gonna guess the third up from the bottom. I don't know how to
Right,
say
that is a good guess. Duodenal jejunal flexure. You do not have to know that name. So cross that one off the list. So far we have names that I do want you to know. Stomach and duodenum. I do not want you to memorize duodenal jejunal whatever, flexure. Don't memorize that one. Okay, what's this one? If this is the duodenal flexure, duodenal jejunal flexure, what is this pointing to? Who's next? Shelby.
Is that, wait, which one again?
This one right here.
Is that the semicolon?
Nope.
No.
So this is part of the small intestine. I'm just going to wrap around when we get to this one. It's the next segment of the small intestine. So we start with the duodenum, and then, Remy?
Jejunum.
Jejunum. What is this yellow, important material that is connecting the small intestine to other small intestines? Sophie?
The mesentery.
That's the mesentery. What is this connection from this part? Well, let's do this one first. What is the next segment of the small intestine? We've got duodenum, jejunum, and ileum. Ileum. Then what's the name of this junction, this connection between the ileum and this next thing? What's the name? I think I gave it to you.
The ileocecal junction.
The ileocecal junction, because this is the ileum, and now we know this is the cecum. The cecum is the first segment of the large intestine. What is this thing dangling off of the cecum? Appendix. Robbie, what's this then? Ascending colon. So if those are some of the words, names to know. So which one? I think we only crossed out this one. Okay, that one you can ignore. Let's play this a little bit more. So here is some more of this. You can cross off Taenia coli. You do not have to know Taenia coli. Okay. We already did the intestine. We already did the ileum. There is a cartoon of the ileocecal junction looking inside the cecum. Ascending colon. We already got that. This is the right colic flexure, which you do not have to know. You can cross that one off your list. What is the name of this structure here? Who's next? Yeah. That's the transverse colon. It's not listed here, so you want to add transverse colon to your list. Okay, Sarah, what's this? Descending colon. So again, you want to add that to your list here. Okay, Abby, what's this?
Sigmoid colon.
Sigmoid colon. Add that to your list. What's this? Oh, who's next? Yeah.
Is that the rectum?
Rectum. Sigmoid colon. Rectum. And who gets anus or anal sphincter? It's the one sphincter you're aware of. Let's see, so here's some of the arteries, but we're gonna see more of them. This is the superior mesenteric artery. And this one here is the inferior mesenteric artery. And we'll talk about the names of some of the others on another
What was the
slide.
difference? Did you say that again?
Superior mesenteric, that's this one right here, and inferior, upper and lower, where they connect to the abdominal aorta. So if I didn't call their name out, we don't have to know. Mesocolon you can scratch off. Epiploic appendages you can cross off. We already crossed off right colic flexure. We're not specializing between the anal canal and the sphincter, just anus. Ileum, appendix, we got. Asemicolon, we got. Superior mesenteric artery, you will need. Tinea coli, you don't need. Tinea coli is this connective tissue on both sides of the large intestine, but you don't have to know it. So that's the gross anatomy. Bobby?
What sort of landmarks do you look for to distinguish it between like the lower of the two branches, like between the mesenteric arteries?
What landmarks between the superior and the inferior
Yeah,
mesenteric?
and then the two that are lower. How do you distinguish between the two of those?
You mean down here? or-
Is that just
I will not ask you to discriminate between those. I do want you to know that the superior mesenteric is over here on the right side of the body and connects to a higher point on the abdominal aorta. The aorta then does the inferior mesenteric. But the inferior mesenteric mostly drains blood on the left side, and the superior mesenteric mostly drains blood on the right side. We'll see some other differences as well. It will be clear. I will not ask you to, so if I point to here, versus pointing to here, I will not make that distinction. Was that the nature of your question? Okay, so now let's turn to physiology. Physiological mechanisms. And I want to start with the hardest one first. Fat. So we talked a little bit about digestion of fat at the very beginning of this section of the course, about how bile emulsifies fat. I use that word in this room, right? Emulsification. So we're going to talk some more about that. So here is a bile salt. It's derived from cholesterol. You do not have to be able to draw a picture of the bile salt. All you have to know is that it's a cholesterol derivative. And it is amphipathic. It has a nonpolar part and a polar part. So it's sort of like, it's not a lipid, but it's like a lipid, and that makes it like a detergent. Because the nonpolar side can interact with the nonpolar parts of fat, and the polar side can interact with polar things like water. And that helps it emulsify and solubilize fat, makes the fat interact with water better. Parts of it interact with the fat, parts of it interact with the water. That's what a detergent is. So when you take, Joy detergent and wash your dishes, you're doing essentially the same thing. It's a better detergent than this. So fat, when exposed to bile salts, where did the bile salts come from? It was one of those functions. The liver. The liver synthesizes bile, which contain these bile salts. So where is the fat here? It's in the intestine. The bile salt is delivered in bile to the intestines. Now they're starting to mix. We form droplets, emulsion droplets, which are small. So it increases this, I mean, it's semi-molecular in size, right? This is a molecule. This is a lipid molecule. This is fat in here. So this is a macromolecular structure of fat and bile and some lipid that is being formed in the large intestine. I mean, in the small intestine, in the duodenum. These emulsion droplets can get further solubilized into a structure called micelles. How many, do you cover what a micelle is in biochemistry? I would imagine Kalani hits that pretty hard. So, one of you who've taken, I want to be accurate here, somebody who's taken biochemistry, how does Kalani describe a micelle?
So are those the ones that form the hexagonal like shapes where you put the tails like outside, and that's the ones that they form, the shapes with 6 sides?
Okay, I didn't know that there was a hexagonal shaping to a micelle. The way I envision a micelle goes like this. We have lipid, head group and tail. We have lysolipid, lipid that has lost a tail, but it still has a head. So lipid, lysolipid. Is that a name that you hear in biochemistry? No. Lysolipid. Lipid, lysolipid. There's some more lipid. Let's just make some more lipid. We have the bile salt. Just gonna draw. There's the bile salt with the nonpolar end facing the nonpolar tails, and then the OH is, out here somewhere, right? So we've got a bile salt. So this structure is a mixed micelle. It's a mixture of lipid, bile salt, lysolipid. It might actually have some triglyceride, depending on what you ate. Remember, some of the fat that you ate is lipid, and some of the fat that you ate is triglyceride. That's fat. And it might have some protein mixed in. That's a mixed micelle. Mixture of lipid, triglyceride, bile, salt, bile detergent, more lipid. And it's forming it is a macromolecular assembly, but it is smaller than an emulsion. And but it's kind of the same principle. But the point is we increase the surface area thousands-fold between a droplet. That's good. We increase the surface area between a droplet, the emulsion, and the micelle. Now there's a lot of surface in contact with the constituents of a micelle. I'll stop moving a little bit. So we're here. If we hadn't had bile, we would be staying as a fat droplet, and that would be causing intestinal distress, right? Then you wouldn't be digesting the fat. The micelles are in equilibrium with monomers of the monoglycerides and lipids and fatty acids. Oh, that was the other constituent. Sorry, one more constituent. free fatty acids. Just the fatty part of a lipid molecule. So the monoglycerides and the fatty acids, just leave it there. The monoglycerides, one of these tails, and the fatty acids is in equilibrium and can form monomers. You could say, wait a second, fats don't dissolve. Well, it's true, they don't like to dissolve. So this equilibrium is favoring the micelle. But there is some exchange of these monomers into solution. It's favoring the micelle, but these monomers still exist. Monomers of the monoglycerides, free fatty acids, even some phospholipid, diffuse into the cells that line the intestine. Remember that the cells that line the intestine have this large surface area that allow diffusion to be efficient. Simple diffusion of fatty molecules, fatty acids, and monoglycerides into the cytoplasm of the intestinal cell. In the intestines, the intestinal cell assembles those monomers into a structure called a chylomicron. Where is it? Here it is. Chylomicron. So it takes these monomers, assembles them back into triglycerides, and then assembles them into this structure called chylomicron, which is then exocytosis. So this is the process that you learned about in cell physiology, just, constitutive. This is not like synaptic transmission that is calcium dependent. This is constitutive exocytosis of chylomicrons. So whenever there are chylomicrons here, the cell says, okay, let's get rid of it, and exocytoses it on the basolateral membrane. Apical diffusion, basolateral exocytosis. Apical diffusion of monomers, basolateral exocytosis of assembled chylomicrons. Now you would say, okay, so those chylomicrons are picked up by the blood, right? Sorry, not right. They are picked up by lymph. So in the villi, the folds of the intestine, is a structure called the lacteal. I want you to know that. The lacteal is a branch of a lymphatic vessel. So this is the lymph. We don't talk about it in this course except for right now. You've heard about blood, of course. You've heard about arterial and venous blood, of course. But we also have another system for drainage called lymph. And now this drainage is important for draining fat from our meal, draining it, and it's going to go into blood eventually. Because this lymph is going to drain into a lymphatic vessel in the abdomen, which is going to drain into the left and the right subclavian vein. And now we're in the blood. But the point is, the chylomicrons are delivered into lymph, not into blood. So these are the epithelial cells that diffused, assembled, exocytosed, and then that exocytosis of the chylomicron is picked up by the lacteal. not by the capillaries of the bloodstream. Is that clear? Yeah.
So, last time when you talked about sugar and protein being absorbed in a different way, so that was referring to the capillaries and then.
That's, we're gonna do that next.
And then the fats are into the lacteals and then they go to
The fat molecules, the chylomicrons that contain the fat. And another name for chylomicron that you've heard of is LDL, low density lipoprotein. So LDL is one type of chylomicron. So chylomicrons go into lacteal, go into lymph, which then go into the venous circulation in the subclavian. Carbohydrate and protein, that is to say sugars from carbohydrate and amino acids from protein, are going to go into these capillaries. Why we evolved it this way, I don't know. Questions about the story of fat digestion, absorption, and delivery to the lymph? Any questions about that story? Carbohydrate is easier, except, well, it's easier, but there are some more details. So we have talked, Sophie?
Sorry, I just thought of it.
Okay.
So from the lymph, like does it go to lymph nodes or what is the fat?
Lymph nodes are junctions of lymph. And so another function of lymph is part of the immune system. And lymph nodes are where, you know, bacteria and the killer cells, you know, the macrophages and stuff that attack foreign invaders get processed and sort of degraded at lymph nodes. This lymph is going to go through lymph nodes, but the nodes are, for this story, are just junctions of lymph vessels.
So where does the fat end up?
Where does the fat ends up? Going into the venous circulation, where it is then going to get circulated around into the portal vein, and it's going to go to the liver for processing into storage, or into fatty acid, or, yeah, one of those two things.
And what is the junction between the lymphatic system and like the capillary system?
There is no junction between the lymphatic system and the capillary. There is a junction between the lymph and the venous circulation in the big vein. This subclavian vein is a major vein. It's not a capillary.
Oh, okay. Thank you.
So if the junction's there, does it go through the superior vena cava?
Yeah, it goes to the superior vena cava, into the heart, gets circulated around the heart, goes all the way through the circulation until it gets to the portal vein, and then goes to the liver. It's a circuitous route. Why this evolved, I haven't studied. It's a pretty good argument against intelligent design. Doesn't make any sense. It must make some sense. It makes evolutionary sense. It doesn't make sense from an efficiency of flow perspective. Carbohydrate. You need a baked potato. Let's say you eat a baked potato with butter on it. The butter follows this story. The baked potato follows, the starch of the baked potato follows this story. You remember that amylase from the saliva and from the pancreas start the digestion of carbohydrate and break them down into oligosaccharides, and maybe break them down into disaccharides, two sugars. you may know that there's the most common disaccharides are maltose, isomaltose, sucrose, which you're familiar with, that's table sugar, and lactose, milk sugar. So ose means it's a sugar, ACE means it's an enzyme that hydrolyzes that disaccharide. So maltase is specialized for the breakdown of maltose. Maltose is two glucose molecules connected by a glycosidic bond. So glucose is maltose, and maltose breaks it down into glucose, two glucoses. I am not going to ask you to memorize the molecular pairings for maltose, isomaltose, sucrose, and lactose. I don't, but just so you know, maltose is glucose. Isomaltose is glucose with a different bond. Sucrose is glucose and fructose. Thank you very much. Glucose-fructose pairing makes sucrose. What's lactose? Glucose and galactose are the pairing for lactose. So you have enzymes, maltase, isomaltase, sucrase, and lactase. They are resident on the apical membrane of your intestines. So amylase is squirted by the saliva and the pancreatic juice into the contents, but these enzymes are resident on the apical membrane. These enzymes make disaccharides, and these enzymes break the disaccharides down into monosaccharides, glucose, galactose, and fructose. And now let's talk about how we get those monosaccharides into our body. This is a story you know, you just don't know it yet. There is a sodium glucose co-transporter in the apical membrane, and this, so you've seen this before. Where have you seen this transporter before? In the proximal tubule of the kidney. It was important for the reabsorption of glucose. Now it's important for the absorption from a meal of glucose and galactose. So this protein transports both glucose and galactose. It uses the sodium gradient as the energy for pumping these sugars from the lumen into the cytoplasm. Sodium-driven co-transport. Then, on the basolateral membrane, we have a glucose uniporter, passive transport. You've seen this before. This looks just like a kidney cell. Maybe. Well, kind of. Epithelium, proximal tubule, kidney cell. Pretty simple. So that's how we get glucose and galactose from the lumen of the small intestine into the lamina propria, where it will get picked up by capillaries. Yes?
So the glucose sodium co-transporter does not recognize how specific galactose against glucose is, so it transports both.
It is.
I see.
It is. It will transport either of these. So this is not both at the same time.
Yeah, but
this is sodium and one of these at the same time. But it will it's not specific for glucose. It will transport galactose. And that's true also in the kidney. It's just that we don't have circulating galactose in our blood. So we don't have to there's no reabsorption of galactose because galactose is converted into blood glucose in the liver before we circulate it around.
Same with the uniporter. It just diffuses galactose, it just diffuses.
Yeah. Same story. Fructose is a slightly different story. Fructose does not use the sodium-driven transporter. It uses a uniporter on both the apical and the basolateral membrane. So it uses a specific uniporter on the apical membrane. You don't have to worry about GLUT5. I am not going to test you on GLUT5 or GLUT2. But I do want you to know that for all three of these monosaccharides, the basolateral membrane has a passive transporter. For galactose and glucose, on the apical membrane, it is sodium-driven co-transport, but for fructose on the apical membrane, it is passive diffusion. So let's talk for a minute about lactose intolerance, just for a moment. So human beings evolved around 10,000 years ago. Some human beings evolved a mechanism for preserving the expression of lactase even in adulthood. So we are born with an enzyme that helps us digest milk, which obviously is very important for the digest. That's our meal for the first year or so of our life. But then, as we start to eat food other than milk, in many of us, the expression of lactase goes away. About 10,000 years ago, as humans started domesticating cattle and drinking milk from animals, a mutation evolved that allowed older children and adults to digest milk. So those of us who drink milk or eat cheese or ice cream without intestinal distress are mutants. We just are. We carried this mutation to allow us to express this enzyme into adulthood. It spread throughout the human population very quickly because obviously it gave a competitive advantage, another source of calories and protein and nutrients. But there are many thousands, millions of people who are not mutants, who can't eat milk or milk products in adulthood because they have downregulated the expression of this enzyme. And so if lactose is present, nothing breaks it down into galactose and glucose. It still stays lactose. This enzyme doesn't transport lactose. It only transports galactose and glucose. If you have lactose in this material here, it's not going to be absorbed. It's a solute. Water flows, diarrhea, intestinal discomfort.
People take certain pills to help them. Is that just like artificial lactase?
It is lactase. It is lactase that has been treated so that it can survive the trip through the stomach and get into the intestines along with the lactose. And then now we have exogenous lactase that will help the body, which dissolves or digests, well, it breaks down the glycosidic bond between glucose and galactose and breaks down the lactose. It's the enzyme lactase. It's just been formulated so that it's not completely destroyed in the stomach.
Is that the sodium potassium use on the basolateral membrane?
Yep. That should be familiar by now.
I did not see the ATP, so I was like.
Yeah, we don't see the ATP hydrolysis, but that's what that is. Yeah, so this is not a sodium potassium antiporter, this is the sodium potassium ATP. Yeah, that's a good question actually. It looks like a sodium potassium antiporter. Okay, I've been talking now for over an hour. Let's take a break right now, just a 5-minute break, and then we'll come back and talk about the protein.